I'm really delighted to welcome Professor Nathwani, whose work I don't understand at all, except that I know that those people who have got haemophilia B and whose lives may well be, positively affected by the findings of his study are going to, this is something that will make a real difference to lives. So I think it's really exciting, and my only hope is that by the end of the evening, all of us might know one more thing. Because I come to these, many of you know me, I come to these every single month, and I always welcome the principle that if I know one more thing at the end than I did at the beginning, it's not bad going. So, welcome Amit, thank you very much, and we're all yours.
Thank you, Jen. Good evening, and thank you all for coming this evening, straight after Easter. So I'm going to talk a little bit about our work on gene therapy for hemophilia B and how the results of this clinical trial that we've done recently impacts on some of the other disorders that we'd also like to treat. So hemophilia, as some of you may know, is a bleeding disorder. And so before we delve into gene therapy for hemophilia, I just thought I'd run very quickly past how blood usually clots. So when we injure ourselves, there's usually injury to blood vessels, and this results in bleeding. The blood vessels respond by tightening themselves to slow the rate of blood loss. Okay? And then what happens next is cells in the blood, called platelets, will form a plug to prevent loss of blood. Now this plug is rather frail, and if it wasn't for the fact that we have what are called clotting proteins or factors in our plasma, this clot would soon disappear and would continue bleeding. But what these clotting factors do is form a scaffold of fibrinous material, as is illustrated here by a scanning electron microscopy. This forms a scaffold that makes this clot really quite firm and prevents further loss of blood and ultimately resolution of this breach of the vasculature. So in hemophilia, a couple of these proteins are missing. So one thing that I need to say about hemophilia is it's a genetic condition. It arises because of mutations, damage, or changes to genes. And these genes essentially are located on the X chromosome. And because us men have only one X chromosome, we're more likely to suffer from these bleeding disorders. So hemophilia A is much more common than hemophilia B and affects 1 in 5,000 males, results from a defect in the factor VIII gene, which means that the factor VIII clotting protein cannot be made. In contrast, hemophilia B is rare and results from a defect in the factor IX gene. And as a consequence of this defect, factor IX protein cannot be made, and consequently, after damage to blood vessels, we cannot form a blood clot. Bleeding continues for longer than usual in these patients. So what are the clinical consequences of this? Well, most patients with hemophilia suffer from frequent spontaneous hemorrhages. So these hemorrhages occur for no apparent reason. It's not because they've knocked themselves against anything or they have had or taken part in excessive exercise. These things just come straight out of the blue. These bleeds usually affect the joints, and so here I'm showing you a picture of a child with hemophilia A who is suffering from a bleed in his elbow. Here's an adult who has a bleed in his knee, and the blood loss has spread from the knee joint into the muscles in the upper leg. And so the consequences of recurrent bleeding in these patients is a deformative arthropathy. So you can see this gentleman who's had several bleeds into this knee joint has a deformity now together with loss of muscle, and therefore his knee constantly, in fact permanently, remains flexed and so he cannot walk without the aid of sticks. Sometimes these bleeds occur in the brain. You can see a large blood clot here in the brain, and these are invariably fatal. So about 10% of children who are born with hemophilia die because of spontaneous bleeds into their head. So the current standard of care, the current treatment actually is very good, and it's very simple. What we do is make the proteins that are missing from the patient's blood and infuse them into the patient. So we can either get these proteins from the plasma, from blood donors, or make them through recombinant protein engineering techniques. So these obviously are much safer because they eliminate the risk of transmission of pathogens through blood products. And so these proteins can be given either at the time of a bleed or prophylactically. In other words, twice or 3 times a week to prevent bleeding episodes. And this treatment is highly effective. It has transformed what is otherwise a life-threatening bleeding condition to one where patients can enjoy a near normal quality of life. And this is best illustrated by Alex Doset, who is a patient with hemophilia A, is a member of the Sky cycling team. In fact, was very, very effective in the recent trials that occurred here in the UK. And this individual, this gentleman, treats himself once every day so that he can participate in really quite professional, aggressive cycling. And he's able to do this without any damage to his joints or bleeding into any of the other tissues. So patients can enjoy a near normal quality of life. So the downside of this treatment is that it's highly invasive. It needs to be given intravenously. Some of you who are diabetics are used to injecting yourself, but into the subcutaneous space, into the skin. But to actually cannulate yourself into a blood vessel is quite challenging for patients. And in kids, this can take several hours each day before the child goes to school. So these frequent intravenous administrations have to be performed 2 or 3 times a week for the lifetime of the patient. This treatment is very expensive, so the cost of treatment for hemophilia B in the UK is about £150,000 per patient per year for prophylaxis. And despite all this, this treatment obviously is not curative because the defective gene in these individuals still remains. They still cannot make functional protein. And so once the effects of what has been injected intravenously wears out, they are still at risk of bleeding. And all patients on prophylaxis have 2 or 3 spontaneous bleeds despite the fact that they're using prophylaxis, administrating this protein on a regular basis. What is most sobering to me is the fact that these protein treatments have been around for more than 20 years. And they're easily and widely available in the UK. In fact, all of our patients in the UK are on some form of prophylaxis. But the majority of hemophiliacs around the world, in fact, 80% of hemophiliacs around the world have little or no access to this treatment, predominantly because of this massive cost of therapy, but also because majority of these individuals live in very rudimentary circumstances where this type of treatment is very difficult to deliver and administer. So what we wanted to do was develop a vaccine type approach that could be delivered once and would be effective for the lifetime of the patient. And this is why we decided to look into gene therapy, because it is a way of trying to fix these genetic problems at its source by deliberately transferring a normal copy of the functional gene. So we're replacing what is a defective gene with a functional gene. And let me illustrate this in the form of a cartoon. So what happens in patients with hemophilia B is they have a defective factor IX gene in the liver. They therefore cannot make normal factor IX protein and consequently suffer from bleeding. Now what we hope to do in these patients is to deliver a normal copy of the factor IX gene using vehicles that are called vectors. And we'll come back to this in a minute because these vectors are generally viruses, and I'll explain why. So we deliver this gene to the vicinity of the liver and then hope that the DNA in there makes its way to the nucleus of the cell, and once it's there, can be used as a template to make normal factor IX. And so once we start making normal factor IX protein, this will give us protection from spontaneous bleeding. So that was our hypothesis going into what was 10, 15 years of work. So what I'm trying to say is gene therapy offers the potential of a cure when protein replacement therapy does not. And our idea was that a single administration of our vector would result in 24/7 endogenous expression, so expression within the patient of the missing proteins, either factor VIII or factor IX, for the lifetime of the patient. We started with the hemophilias, not just because I'm a hematologist, but because they are well suited for early phase proof of concept studies with gene therapy. And the reason for this is that we know that they arise from a defect in a single gene. So compare cancer. Cancer, as some of you will know, is a multi-genetic disorder, whereas we know that hemophilia A results from a defect in the factor VIII gene and hemophilia B from a defect in the factor IX gene. Other genes are not involved in the pathogenesis, so this makes it very, very simple for us. Additionally, the therapeutic goal is modest. And what I mean by this is that these proteins actually circulate in the plasma in minute quantities. And years of clinical experience has taught us that if we can get a rise in either the factor VIII or the factor IX protein level to just above 1% of normal, so around 50 nanograms, so we're talking about minute quantities of protein, this would be enough to ameliorate the bleeding phenotype. So this would change the phenotype considerably in a patient with severe hemophilia. Additionally, we have assays in all the routine coagulation laboratories which can be used to assess efficacy. And also important is that tight regulation is not required. So compare this to diabetes. So if we were introducing a gene for insulin, for example, we would have to make sure that it is very tightly regulated depending on the glucose levels in our blood, which is then dependent on the amount of exercise and the kind of food we eat. Whereas for factor VIII and factor IX, a wide range of protein levels are likely to be efficacious and not toxic. So these were important attributes to why we decided to go with gene therapy of hemophilia as opposed to other diseases. Now, before I discuss our trial and the results of the trial, I want to introduce you to this term vector. So what we've done is used viruses to deliver our genes. And this is because viruses have evolved over time to become highly efficient at delivering their payload, which is usually DNA or RNA, to their target cells. And we wanted to take advantage of that so that we can develop a very simple route of delivery for our genes that we wanted to use for correcting hemophilia. Now, before you become horrified, what we do with these viruses is we take a viral-type virus. And the first thing we do is disassemble the viral genome. We basically pull it apart, get rid of the parts of the DNA that are important for virulence. Okay? We replace those parts with our gene of interest. So this could be factor VIII or factor IX. Other parts of the DNA are then put into separate plasmids so that we can then introduce these separate plasmids into cells in culture in the laboratory to make our recombinant viral particles. And what I'm trying to say to you is that the recombinant viral particles that we use for gene therapy bear little or no resemblance to the wild-type virus that we had started off with. What we've done is modified the capsid so that it targets our cell of preference, and we've made it substantially safer by taking out all the components that are important for virulence and division of or replication of this virus in the target cell. So the virus we use for gene therapy is called adeno-associated virus. This is a non-pathogenic but single-stranded DNA-based parvovirus. It's in fact a very tiny virus. It's the smallest virus that we know of. The reason why we chose this is because 70% of you in this room will at some stage been infected with AAV, as I'll refer to it from now on. But it does not cause disease. So unlike adenovirus or the influenza virus, where some of you are coughing with flu, AAV is completely benign. It does not seem to cause disease. What it does is it piggybacks itself with adenovirus or the influenza virus and gets into the target cell. And once into the target cell, it uses these helper viruses to divide and replicate, but it does not cause disease. We were interested in it because of its great safety profile, because it's not associated with any pathology, but also because we could engineer these viruses very easily. So what we can do is take out the wild-type genes, the rep and cap genes, and replace it with our gene of interest. So this is our factor IX gene under the control of a liver promoter. And what we have been able to demonstrate with these AAV viruses is that a single administration of this virus, either in the muscle or in the vicinity of the liver of animal models, including non-human primates, results in long-term production of the factor IX protein. And we have animals that are alive now 10 years following a single administration of this vector that are still making sufficient quantities of the factor IX protein that allows these animals to have normal hemostasis so they don't bleed when they're challenged. And importantly, these viral genomes are maintained episomally, so outside of the chromosome, therefore reducing the risk of inducing cancer. For our clinical trial, we didn't use AAV2 because this is very common in humans, but instead we used AAV8 because very few of us have ever seen AAV8 before, and therefore we don't have immunity to AAV8, whereas the majority of you will have some level of immunity to AAV2, and so administrating this would result in neutralization and no infection. Additionally, we made these vectors much more potent by introducing a second copy, because remember, this is a single-stranded virus, so we needed to introduce a second copy of the factor IX gene, because most of our genes exist in 2 copies, so we had to introduce the second copy. And finally, we were able to administer the virus into the peripheral circulation, because AAV8 has a remarkable ability to home to the liver, which is where factor IX is normally made. So it doesn't matter where you administer the vector, you can administer it into the subcutaneous space or into the muscle or into the blood circulation, it will find its way to the liver. And so we use this to our advantage to develop a simple mode of delivering the vector in patients with a bleeding diastasis, so we could avoid the need for an operation because we wanted the vector to make its way to the liver. So again, just to give you an idea of how long these clinical trials take from conceptualization to actually going into the first patient. And so the vector design and testing of this self-complementary vector was completed by 2005. It had taken us 6 years to get to this point. We then started developing a method so that we could manufacture this vector in a method that was suitable for use in humans. We needed to get approval from the ethics body in the UK called GTEC, and then the lawyers came into play. So this was really a very, very difficult time for us because the vector was made in a pharmaceutical type facility at St. Jude Children's Research Hospital, which is where I'd started this work some 10 years before. They were at that time the only facility to have this pharmaceutical type facility or laboratory to make vector for use in humans. But obviously the lawyers wanted to make sure that each institution, UCL and St. Jude, had relevant indemnities in place because they didn't want to take responsibility for anything were things to go wrong. And so these negotiations took 2 years, and ultimately we were all going round and round in circles, whilst the lawyers, as always, were making a huge amount of money. Ultimately, we had funding for this trial from the MRC in the UK and the NIH in the US. And the vector was made in this GMP facility at St. Jude Children's Research Hospital at a cost of £850,000 for 10 patients. The trials were approved by the regulators here and in the USA, and the first patient was dosed in 2010. So this was a phase 1 study, and as is required by law, the objective of these phase 1 studies is to establish the safety of our intervention. In the UK, we're allowed to look at efficacy, but in the US, efficacy is a secondary criteria. It cannot be a primary objective. And so in this study, our vector was administered as a simple peripheral vein infusion into patients with severe hemophilia B. These individuals had less than 1% of normal factor IX protein in their blood, and we did this without giving them any immunosuppression. The patients who had pre-existing immunity to AAV8, in other words, if they had been infected in the community because of a wild-type virus, these patients were excluded, as were patients with active infection with hepatitis B, C, or HIV. So there were 3 study sites, but in fact, all of the first 6 patients recruited to this trial were recruited at this institution in my hemophilia center. The patients here have very good treatment, and so we elected to go through an elaborate consent process because we wanted them to truly understand the risks that they were taking. And remember, this was a first-in-man study. Nobody had infused AAV8 into the systemic circulation of patients with hemophilia. And so writing the consent form for the patients was really quite difficult, but an interesting time. And as I was doing this, I was invited to a party in Memphis, Tennessee, which is where St. Jude is based. And this was on a Sunday in the afternoon. And whenever I think about risks that we all take, this picture always comes to mind. So you can see that my friends and colleagues were having a great time in this paddle pool on a hot Sunday afternoon, enjoying their local beverage and also listening to music. This CD player was powered by a cord which is being held in place by plastic flip-flops. And you can see that the risk of electrocution is substantial. But these well-educated physicians at the Children's Hospital were prepared to take this risk. This is not to say that one needs to mock the risks that we force our patients to take. In fact, our strategy was to try and minimize risk as much as possible. But what we wanted to do was be completely honest with our patients. And so we were very clear in the patient information leaflet that the risks were largely unknown. And in fact, that there was risk of damage to the liver. There's the risk of formation of an inhibitor, which means that they would become refractory, would not be able to treat themselves with the factor IX protein that they had used up to then. That there was a possibility of developing hepatocellular carcinoma. And of course, because this is a virus, they would be sensitized to AAV8 for their lifetime. So we made our patients go through 3 elaborate consent processes, and in fact, ultimately even hired an independent ombudsman. He was a member of the Hemophilia Society here in the UK, a well-educated individual whose job essentially was to talk our patients from participating in the clinical trial. We really wanted to see that they truly understood the risks that we're taking. So I'm pleased to report that despite all of these endeavors, all the patients we approached for gene therapy were in fact determined to take part in this program, which is a testimony of what they think is the current standard of care for them like at the moment. And to just make sure that we didn't do what was done at Northwick Park, we decided to treat 1 patient at a time and follow this patient for at least 6 weeks. So if toxicity were to occur, it would be limited just to 1 patient, not 6 patients as was done at Northwick Park. So rather conservative in our approach, 6 patients were recruited. The first 2 were given low dose of vector. The second lot were given the intermediate dose, and the last cohort high dose of vector, which is 2 × 10⁻12 vector genome per kilogram. When we started out, We thought that the older patients would be more interested in gene therapy than the younger patients because they would want to do this for altruistic reasons, because they know that their daughters are carriers and their grandchildren will invariably have hemophilia. But in fact, to our great surprise, the majority of patients who participated in this clinical trial were rather young. These individuals had a variety of mutations in their factor IX gene and were on preventative prophylactic protein replacement therapy either once a week or 3 times a week. And despite this, all of these individuals reported breakthrough bleeds occurring at least 1 to 4 times a year. And all these individuals had some damage to their joints, which included the ankles, knees, elbows, and shoulders. So what I'm showing you here are the vital signs of the first patient enrolled to our clinical trial. So in green is the pulse, in red is the temperature, and in blue is respiration. And you can see that after administration of the vector in this individual, there was a slight increase in his pulse, which went up to about 75. But I can guarantee you, ladies and gentlemen, that his pulse was substantially lower than mine at that time. This was the first time we were administrating this vector, and mine was off the scale here. So in fact, this is our gentleman receiving the vector. You can see that the vector is infused over a period of 1 hour through this CAD pump into his peripheral circulation. He's really very comfortable. In fact, he was so bored that as an IT specialist, he was frustrated that I couldn't take his picture on this momentous event using my BlackBerry, and decided to try and fix my BlackBerry whilst receiving this infusion. So this in itself is quite remarkable, and we were really quite surprised about the stability of these measures, including temperature, because when you look at where we could pick up the virus, so after injection or infusion of the virus into the peripheral circulation, we could pick up the viral genome in the blood, which is not surprising, and this lasted for about 3 weeks. But we could also pick it up in the stools, in the urine, in the saliva, and also transiently in the semen. But all of these signals disappeared within 3 weeks of administrating this vector. So remember, this is just a single one-time infusion of the vector into the patient. And this is just liver enzymes of some of the patients, and you can see that when compared to pre-treatment levels, the liver enzymes essentially remained within the normal range. So this is the upper limit of normal in these patients. What I'm showing you here is the factor IX level in the first subject enrolled into our clinical trial. So he had his protein infusion, and then we took blood samples and measured factor IX levels over time. So what we saw initially was a washout of this protein that was infused prior to administrating our gene therapy vector. But instead of the protein levels dropping to less than 1%, in him, they stabilized at 2%. So this was really quite surprising, because all of the work that we'd done in animals had predicted that this dose would not be efficacious. So we were not expecting to see any benefit for these patients at this dose level. And so when we saw this, we were really quite surprised. And for about 6 weeks, we didn't tell anyone, because we didn't believe it ourselves. But you can see that his factor IX level has remained at between 2 and 3% for a period that now extends up to 4 years after this single administration of vector. He used to treat himself 3 times a week. He's stopped prophylaxis, but has not suffered any spontaneous hemorrhages. He has required some treatment with factor IX protein, but this is usually after he's involved in a bar fight. He has a strategy as an IT specialist of working hard and playing hard. And all of these events mark either Easter or the New Year. So he's out celebrating and then comes into the hemophilia center with a black eye or some fall, and we're then obliged to treat him. And you can see that his factor IX usage, so prior to gene therapy, he was infusing something like 4,000 international units per kilogram per year of factor IX every year before gene therapy. After gene therapy, you can see that despite these traumatic events, his factor IX usage has dropped substantially by about 76%. So this was really quite a surprising outcome. So we then moved up a dose to the intermediate dose level. In subject 4, you can see that his factor 9 level, which was less than 1% before gene therapy, stabilized at between 3 and 4%. This individual didn't have as severe a phenotype as the first patient that I showed you, and in fact, he could get away with treating himself once a week, usually before he played football or cricket. Okay, following gene therapy, he's not had to do that. He's able to play football and cricket without administrating protein. Again, these protein infusions, which are 4 infusions over a 3-year period, were given to him because of traumatic injury. When he hit himself or fell himself, he got himself a large bruise, and we had to treat him on those occasions. Moving up to the high dose level, you can see that in subject 6, his factor 9 levels stabilized at between 8 and 12% after administrating our vector. This individual was on 3 times a week, but after gene therapy, he was able to train and run in a marathon without any prophylaxis. So he did not give himself any protein therapy whilst training or running in a marathon. 3 days after running in a marathon, he was reviewed in our hemophilia center, and we saw a slight rise in his liver enzymes. So it went from 15 to 30, so this is still within the normal range, and was associated with an increase in another enzyme called LDH. And this suggested to us that the strenuous exercise of running in a marathon was the cause of these abnormalities that we were seeing in his blood. Remember, these patients are under the microscope, and we're looking at every parameter we possibly can. But when his factor 9 level dropped to 4%, we thought that this might be an immune response to the viral vector, and started him on immunosuppressive regimen with prednisolone. So this is prednisolone, 60 milligrams. This is a dose that I take when I have a flare-up of my asthma. He was on this treatment for about a month, and you can see that his liver enzymes have gone back to normal. His factor 9 level has stabilized at between 4 and 5% of normal. Okay, so he was on 3 times a week treatment. He's stopped that. He's been able to stop that for almost 3 years. In fact, he's not required any treatment over this time. And his factor 9 usage, which was 3,000 international units per kilogram per year, has gone down to 0. This is what he thinks of life after gene therapy. I'll give you a couple of seconds to read what he feels. So the high dose level is really the business end of the clinical trial. And so we recruited a few more patients at the high dose level, and again you can see that in this individual who used to treat himself once a week and still had 19 bleeds a year, his factor 9 level following gene therapy has stabilized at about 5%. He's not had any bleeds, he's not required any treatment. His liver enzymes have remained in the normal range. Another patient in the US, they don't use prophylaxis very much in the US. This individual was getting more than 1 bleed a week. I mean, he has a very, very severe phenotype. He still was on on-demand treatment because of their healthcare system. And you can see that his factor 9 levels stabilized again at about 5%. His liver enzymes have remained in the normal range. He has not required any treatment with steroids. So he used to have 54 bleeds a year. Following gene transfer over a period of 1 year, he's had 0 bleeds, no bleeds whatsoever. So his life has been transformed. So in summary, ladies and gentlemen, this is the first successful gene therapy trial for hemophilia B in the world. I've shown you evidence of endogenous expression, so this is expression from the patient's liver of factor 9 protein. This is a protein that was missing in these individuals. This protein has now been made for a period that extends up to 4 years following a single administration of the vector. 5 of the 7 patients of the 10 we've recruited who were on protein replacement therapy as prophylaxis and have been able to stop this without spontaneous bleeding and with significant improvement to their quality of life. What is really quite interesting is the net saving just from a reduction in the usage of factor 9 so far amounts to £1.5 million and is accruing on a daily basis. The treatment is relatively safe. I mean, we have seen asymptomatic inflammation of the liver, but this is really mild inflammation, the kind of inflammation that we would all suffer from after a glass of wine or a couple of paracetamols. But you can see that a short course of steroids is enough to stop this inflammation. So I think, ladies and gentlemen, you'll agree with me that these preliminary results are highly encouraging and suggest that gene therapy can change the treatment paradigm for hemophilia B. Just in case you think I'm going to stop waffling, I'm going to move on to the next target. But before I do this, how long is it going to take before gene therapy becomes a medicine that you can buy not over the counter, but from a pharmacy? And so after our success, which we published in the New England Journal in 2011, several groups have become really quite interested, with big pharma now interested in gene therapy. So I think it's going to be a maximum of 10 years before this product becomes commercially available. We've elected not to use gene therapy in children for the moment because we want to see safety data over 5 years before we go into kids. But you can imagine that this is an inherited disease. I showed you a picture of a child with a bleed in their elbow. The maximum impact of gene therapy, in fact, is going to be in kids, not in adults. And so we and our partners at St. Jude Children's Hospital are itching to get into kids, but we want to make sure that we have sufficient safety data to support this. The current cost of making vector in our back garage is about £30,000 per dose, but this will drop substantially with industrialization. And if the drug companies are going to be sensible, which is really a pie in the sky and my aspiration, but if they're going to be sensible, then they would use a model that really is going to be of great appeal to the NHS and to the developing world, where you pay a small amount in comparison to what we pay for protein concentrate for as long as gene therapy works and is maintaining factor IX levels above 1%. This is a win-win situation. Patients benefit, the health system benefits, and the company makes really quite a significant amount of money. But you can imagine that if you leave this entirely to companies, then they're going to price this at several millions because they know that this is a one-time treatment and they want to try and make as much out of this as they possibly can. So the next question was, can we make gene therapy work for hemophilia A? Hemophilia B we started with because it was convenient, but hemophilia A is 5 times more common, as I've mentioned to you, and so the treatment need is obviously greater. But the problem is that the factor VIII gene is also 5 times larger than factor IX, and trying to package these into these small viruses is quite a challenge. But more recently we've been able to do this. We've developed what I call variant 3, which is in fact more efficient than the wild-type virus, wild-type factor VIII gene, and you can see that this evaluation in mice shows that our engineered form of the factor VIII gene is at least 10 times more potent than the wild-type gene. When we put it in hemophilia mice that have a bleeding tendency because they are missing the factor VIII gene, then we get substantial levels of expression of this variant 3 in the hemophilia mice. And then if you challenge them by clipping their tail, they're able to form a blood clot very, very quickly, whereas animals that have not had gene therapy continue to lose blood at a substantial level over a period of time. So this new vector that we have developed seems to protect hemophilic mice from bleeding following a hemostatic challenge or an injury. So these bioengineered forms that we can make in the lab of genes can have higher potency and are small enough now to be able to be packaged into our small AAV vectors. What was really quite surprising is when we did a dose-finding study to try and work out which would be the right dose to use in humans, the same doses that were effective in the hemophilia B trial are likely to be effective in hemophilia A. So this innovation, which we've now licensed to a company called BioMarine, has enabled us to feel optimistic about the prospects of gene therapy for hemophilia A. And we hope to start a clinical trial next year here at the Royal Free in patients with severe hemophilia A, again using the same model that we use for hemophilia B, looking at 3 different dose levels, assessing safety and efficacy in these individuals over a period of time. So one of the reasons why we work with these rare monogenetic disorders where only one gene is mutated or abnormal is because it allows us to establish what we call proof of concept. So it allows us to establish in a relatively simple system whether or not gene therapy is going to work. But of course, we want to use gene therapy for a variety of more common conditions such as cancer, such as diabetes, such as heart disease. And so we began to ask the question whether we could use AAV vectors to target hepatocellular carcinoma. As some of you will know, this is a special interest of the Royal Free. We have a very large, active, pioneering liver unit here at the Royal Free. Hepatocellular carcinoma is the 5th most common cancer worldwide. There are more than 500,000 new cases annually. Surgical resection or liver transplantation works, but only in a small number of patients where the disease is diagnosed early. So if the disease is picked up when the tumor is less than 3 centimeters, then there is a good chance of cure and success with surgery or transplantation. But in the majority of cases, the cancer is indolent until it gets beyond the size of 5 centimeters. And so most of these patients present with advanced hepatocellular carcinoma, which we know is resistant to chemotherapy and radiotherapy. So new small molecules that you may have heard about have had some impact. So a drug called sorafenib has improved survival by 8 weeks. But sadly, ladies and gentlemen, most of the patients who have advanced hepatocellular carcinoma are not alive at 5 years following the diagnosis. In fact, the majority would succumb to their disease within a couple of years. And so you will agree with me that there is a pressing need to develop new therapeutic approaches. So why use AAV vectors? Well, we know, and I've shown you in our hemophilia B patients, that these vectors are relatively safe. And I've also mentioned to you that these vectors have a remarkable ability to home to the liver. So it doesn't matter where you administer it, they seem to find their way to the liver. Mechanism of this we don't understand, but we like that fact. And so therefore we began to wonder if we can use these 2 properties to develop a therapeutic approach where we can deliver therapeutic genes selectively to metastatic, in other words, advanced hepatocellular carcinoma, without causing any toxicity to normal tissue, including the normal liver. Now I'm going to go through some animal data and try and explain what we're trying to do here. So many of you will have seen firefly, and you know that in the dark it glows because it contains an enzyme called luciferase. So we can take the luciferase gene and put it into our AAV vector under the control of a constitutively active promoter. So this gene will work anywhere in the body. When we inject this into the tail vein or within the portal circulation, so within the liver circulation, then the only organ that lights up using a camera that can pick up luciferase is the liver. So you can see that even after tail vein administration, where the vector will go to the heart, to the lungs, other organs, and then the liver, we still see that most of the expression is coming from the liver. So this was nice to see. So the next question was, will we also see expression in liver cancer that has spread? And to develop this model, what we did was we put a lump of cancerous tissue into the flank of a mouse, okay, and then injected the vector into the tail vein. So where we had injected hepatocellular carcinoma cells into the skin of the mice, you can see that these cells light up with luciferase. So in other words, the vector has reached the cancer and the gene is able to express itself in the cancer. But if you used another cancer cell, such as neuroblastoma, and you can see there's a large lump of neuroblastoma here, then that does not light up. And what this taught us was that AAV can preferentially transduce the liver, but also hepatocellular carcinoma cells. So this made us believe that we could selectively target hepatocellular carcinoma cells. Now, if you're going to deliver a therapeutic gene to cancer, you want to make sure that it's not expressed in normal tissue, like normal liver. Otherwise, the normal liver will be destroyed. And so the next set of experiments were designed to see if we could switch off expression in normal liver, but keep the expression on in the tumor. So we wanted to keep the expression on in the tumor, but switch it off in the normal liver. And so what we did by molecular engineering, so we put another gene in here, which allows us to do this. So you can see that by introducing this switch off gene, we were able to switch off expression in the liver of this mouse. But the expression in the tumor is still there. So we now have a potential to selectively deliver our genes just to cancer cells. At the moment, there is no other treatment that can do this other than antibodies. Okay, so when you give chemotherapy, these drugs, as some of you will know, will go all over the body. But what I'm showing you here with our AAV vector is that we can find tumor cells that are spread outside of the liver, selectively transduce them, and express our therapeutic genes in these metastatic tumors. So we then used a therapeutic gene called herpes simplex thymidine kinase, and I won't go into the detail, but again, using non-invasive imaging, so these animals do not need to be killed. We can use PET type or CT scan type imaging to see where the gene has reached. And you can see that with our molecular switch, the only place where we can see the gene expressing is within the tumor. Without the switch, you can see that the liver is efficiently transduced. And this is just a control animal with no vector. And so when we gave these animals, this is an enzyme that activates a drug called ganciclovir. And so when we gave these animals ganciclovir, what we saw was the tumor in the flank stopped growing. But in these animals, because of expression of the enzyme in the liver, these animals didn't survive for more than 2 days. The control animals, the tumor continued to grow over time. So this told us that with our special enzyme and ganciclovir prodrug, we could retard the growth of the tumor. In fact, in an elaborate experiment, and I won't go into the detail, you can see that control animals had substantial quantities of liver tumor in the liver and in the spleen. But after treatment with ganciclovir, we were able to clear most of the tumor in these animals. So we hope to test this vector in clinic, again at the Roll-Free in 2016, using essentially the same schema that we'd used for hemophilia B. So ladies and gentlemen, I've shown you today a diverse range of targets for gene therapy. We have clear evidence of clinical success in hemophilia B, and we hope to use this approach next year in hemophilia A, and then ultimately in hepatocellular carcinoma. But these are the other diseases, and I didn't have time to show you data from these other targets, but these are the other diseases that we are interested in using gene therapy for. And if we can make it work either for diabetes or hypertriglyceridemia, then you can imagine that this is going to have a huge impact to the medical community. So a special mention to our patients. Now, for a long time, we had believed that our patients truly didn't understand the risks that we're taking. Except that after I finished talking to the first patient and before he had his vector administration, he pulled out a document from his coat pocket and asked me to sign it. What he had in his hands was his will. And he wanted me to countersign it so that he could give everything away to his girlfriend. Of course, I refused to do that because in 6 months' time, he separated from his girlfriend. But what this taught me was that actually I was stupid, and my patients truly understood the risks they were taking. And I had just not appreciated that they were really very, very smart and understood that what they were doing was to advance medical science. And so without their help and their bravery, we couldn't have made the progress and success that we've made. You can imagine that these kind of studies require the active participation of a large number of individuals, and there are too many people for me to thank personally. But my colleague in the Hemophilia Center, Ted Tuddenham, was instrumental in recruiting some of these patients. And our collaborators in the United States, Andy Davidoff and Art Nienhuis, continue to support us with this clinical trial. We've received generous supports from a variety of research councils and charities in the UK and have licensed several of our products to industry recently. So with that, I'll stop and be happy to take any questions. Thank you very much for your attention.